Folks, I'd now like to introduce our next two speakers who will be doing a joint presentation. First of all, I'd like to introduce Associate Professor Qifeng from the South Australian Research and Development Institute. Qifeng is the Principal Science of SARDI's Inland Waters and Catchment Ecology Program. She's currently leading the Living Murray Fish Monitoring in the Coorong and the Food Web Research for the Healthy Coorong, Healthy Basin Trials and Investigations Project. Joining her is Professor Sabine Dittmann, also from Flinders University. Sabine is a Professor for Marine Biology at Flinders University and a nationally and internationally renowned expert on coastal ecosystem ecology. Sabine has worked extensively in the Coorong since 2004, where she knows almost every worm by name and focuses on the interface between research, environmental management and policy. I welcome you both to this lectern. Where am I looking? Here. Sorry, I've got very bright lights in my face. Qifeng, for you first.
Thank you, Nicole. How do we operate that? Just click that.
Big button is, yep.
Sure, yeah. All right. Great to see you all. Thanks for the opportunity. I'm Qifeng from SARDI. And I think it's nice timing that it's close to lunch. We talk about how important food resources are. Yeah, so thanks. So I'm going to focus on talking about the Healthy Coorong, Healthy Basin trials and investigations regarding the Coorong food web dynamics. So I'm delivering the first part, and Sabine will join me to talk about the second part. And before that, we would like to thank a host of other researchers, like other program Luke and Michelle presented. Heaps of other researchers contribute to this from Flinders Uni, SARDI, and LA Uni. And particularly, I listed a few task leaders. So George Jaitas, Ryan Baring, and Simon Goldsworthy. Yeah, unfortunately couldn't be here due to other fieldwork commitment. So talking about food web, what is a food web? It's basically a map about feeding relationship between organisms in the ecosystem. And it's showing that how the matters and energy flow through. And overall, that maintaining in the dynamic Coorong ecosystem is very critical about maintaining a functioning and resilient food web. But as other colleagues presented, there are many years that, due to a range of reasons, many about the water resources lacking, is leading to a huge impact and deterioration in the Coorong ecosystem, as you all witnessed that. And therefore, that this clearly show that South Lagoon, the ecosystem change, you got an overly simplified food web in that system. So that basically reduced not only the overall food resource diversity, but the resilience of the ecosystem. So what our research is aiming at is to better understand the food web dynamics and with a few key questions in mind. So those are our key research objectives there. So first of all, you need to understand about what are the key food sources and how important they are for the key species. And here we targeted at key water birds and also fish species. And then second part is that we wonder that what are the key ecosystem drivers, how they influence the food resources availability out there, how diverse they are, how abundant, what is the biomass, and then the bioenergetics, what is the nutritional value or energy content of those food to support the high-level organisms. And so also that overall that we want to bring all this knowledge and data together to be able to develop a quantitative food web model. So you can use that to assess all the environmental change or management interventions on how that affect the overall food web response. It's a very complex system. But ultimately, why we do that is we want to use all this information to inform the restoration of a functioning food web. So when I said a functioning food web, there's always a food web there, but we want to probably direct the energy flow and align that more to the values that we recognize, such as the Ramsar plan, and overall, for the community health and environmental health for the system. So the general approach that this large food web project we took is that we have 4 key activities. We also call it tasks. So first activity is desktop, and we did a thorough review and synthesis of existing data and knowledge, and also trawl through the conceptual models. So what we already know and identify the knowledge gap. And then we got into that task 2 and 3, which is serious on-ground empirical studies combined with the lab work. So first to look at the diet studies, focus on water bird and certain fish species. And the third one is looking at key ecosystem components. So Michelle talked about primary producers, but here we more focus on zooplankton, macroinvertebrate, and fish. So looking at how that the dynamics in the Coorong by sampling along the Coorong salinity gradient, not just zooming to the South Lagoon, but because there's a natural salinity gradient along the Coorong, which help us to look at how different environmental parameters such as salinity affect on these biota. And then I think overall that the last one is a desktop study. We brought back all the information, including the earlier data, bring them together to basically develop a quantitative food web model. So I'm going to go through the first 2 activities. The first one is review and synthesis. What we found is that for the Coorong fish species, through this literature, the diet study, we got fairly good understanding about most of the fish, what's their diet and the relative importance. And so basically that these fish got a variety of feeding modes, and very importantly that benthic invertebrate are a very key component of their diet. And we also found some knowledge gap for some fish species, such as lagoon goby, which is a very small prey species, but it's very important for the ecosystem. There's not much knowledge there, so we identify the gap. And also, we found that for the water bird species, the knowledge is really sort of patchy and lacking, or it's really sort of dated, like 1970s. So that's there. So I think further study is required. Now, another important thing is that we synthesize the data and information for those key biota, like zooplankton, macroinvertebrate, and fish. A lot of information collected over multiple decades, and a chunk of data is from the Living Murray program that we got all these annual monitoring, and it's really thankful that it's lasting for 15 years and it's contributing to this overall long-term understanding throughout that different hydrological conditions and how the flood and drought impact on all these food web components. In addition, we also trawl through the conceptual models, and some conceptual models through me and my colleagues' research, we already kind of developed a conceptual understanding about the broader impact, like what happened during the millennium drought regarding the food web along the Coorong in a conceptual, semi-quantitative manner. But then during the flood, how the system changed. So all these things, it's helping us to create a range of matrix, including the diet matrix that is critical needed for developing the quantitative food web model, but also with some drivers and understanding about how key components change and they will affect the food web relationship. So in terms of their activity 2 about the targeted diet study, so my colleague George, he used that a traditional gut content analysis method to basically investigate these small body fish, so lagoon goby, what is their diet. And then he also worked with the bird ecologists from LA Uni, so Tom Prowse group, to collect a lot of bird poos, so the scat sampling, and that one we used the DNA meta-barcoding, so using DNA to analyze what are the contributing diet to these water bird components. And we focus on the wader species, so shorebirds, and also look at waterfowls, so ducks. So what we found is it's interesting that this diet of lagoon goby, and in the Murray Estuary and North Lagoon, they were almost exclusively that focused on amphipod, so these little bugs, macroinvertebrate, and providing them nutritious food. And then for the shorebird diet, and for sharp-tailed sandpiper, redneck sting, and red-capped plover, in the South Lagoon, the diet is constituted as chironomid larvae, because it's only chironomid larvae could tolerate that hyper saline water, and it's only that choice. But very interesting, in the North Lagoon, so we look at closely about sharp-tailed sandpipers scat. So when there's more diverse food available, then their diet component is diversified. And so they got chironomid larvae a bit, but a lot of amphipod and worms. And it's very important to mention when this choice of prey are available, and these birds, they actually prefer amphipods rather than chironomid larvae. Now for the waterfowl diet, so chestnut and grey teals, not surprising we found that rupia is dominating their diet, but they also feed a range of other vegetations. So a very important key message is that for shorebirds and fish, in the Coorong and I think other places also, abundant and diverse prey assemblage really benefit them. And also that they need, for shorebirds, they need suitable habitat and feeding ground. And for fish, of course, that you need to have favorable condition in the Coorong in terms of salinity, water quality, and also that connectivity. Thanks. I'll leave it here, and I'll invite my colleague Sabine to talk about the next few fantastic work. Sabine.
Yeah, thanks, Qifeng, and hello, everybody. So I'm now continuing, and now we know sort of what the different species are foraging on, where do they actually find their food, and what are some of the environmental drivers that determine where the food is in highest abundance, and also what's the further nutritional value of it all. So this slide here has got already a little bit of a summary of the key points that came out of these investigations. We did look at the zooplankton, the fish, and the macroinvertebrates, and they all showed roughly a similar pattern. And that consisted out of having or following that gradient in the Coorong that you heard about before, that you've had the highest diversities and abundances and biomass in the estuarine section, where salinities are lower, and the further down in the South Lagoon, it tapers out and you've got less of them. And I go more into the environmental drivers in a minute. And what it means for the higher trophic levels is that the choice of prey available is much lower in the South Lagoon than it is in the northern section. And that is also available as a report on the Goida website. So one of the environmental drivers we of course looked at was the flow. So on the chart that you see there for the macroinvertebrates, the blue indicates the flow of water coming over the barrages, and then you've got with the dots there the abundances of the macroinvertebrates in the different regions, the Murray Estuary, the North Lagoon and the South Lagoon. And already when the higher flows commenced in, here in the middle of 2000s. Then the abundances, especially in the estuary and in the northern North Lagoon, started to increase. And towards the end of 22, not all of the dots are in there, but also from the TLM long-term monitoring from the end of 21, we know that the abundances were then getting up here in the estuary. So there was a real response to the higher flows in the abundances in there. And for the fish, also especially in the estuarine region, you see that they're in the middle of the year in June here, and then towards the end, the abundances were getting a little bit higher. Again, most of the response to be seen in the estuarine region. And for the fish, also notice the differences in the overall scale bar here, where the abundances were much higher in the estuary than further in the lagoons. So flow definitely has got a big benefit, and we've seen that also after the recent events now with the big flood that came through, that we have seen abundances that for both macroinvertebrates and fish we haven't really seen for a long time. So water matters and is doing a good thing. The water also lowers the salinity. And from our long-term studies, and these plots here are just for the data that we've got from the trials and investigations, we've done just simple scatter plots for the individual numbers and also the biomass against the salinity. And just for over all of the species of macroinvertebrates, you see that as soon as salinity sort of roughly goes about 50, it really drops sharply. So and we've done other analyses that are sort of corroborating that threshold there in the salinity. And also for a lot of the fish, where you've got, again, the salinity gradient down here, and the blue line, again, shows about 50 salinity. So you see that apart from the smallmouth hardy head, most of the abundances of the fish are dropping when the salinities are getting too high. That's why we've got that gradient and the pattern that we've described before. And so for the invertebrates, I should point out that chironomid larvae are also the only ones really that are accounting for these higher numbers here. So they can occur in a much wider range of salinities. But in most cases, if we are not bringing the salinities down to a lower level, as we've now seen being the benefit of the big flood that we've had, that the South Lagoon is starting to get recolonized. So we really, as Adrienne also pointed out, for the further ambitions with the e-water, try to really keep salinities as low as possible for as long as possible, so that we've got the benefit of having higher abundances and higher biomass for the food web, but also for the other processes that Luke talked about, for then really denitrifying the sediment, the MBO conditions. And so this is another way of looking at it. So we also measure not only how many individuals there are per unit area or per catch per unit effort, but we also looked at the biomass, which is an important variable that goes into further food web modeling. And this is a plot here where the pie charts really, and you've got some scales on there, are showing you sort of here how much biomass you have. So that's wet and dry mass. And then you've got the different colors of the different pie components there, showing you different trophic groups as they were used for the modeling that we then also did. And you are seeing again on this plot that in the estuarine section, sort of this is about Gulba here, sort of in the northern section, there, during the time of the trials and investigations, we've had the highest biomass of fish and macroinvertebrates. And also you've got sort of most different colors there in the pie charts. So quite a different composition of prey items available there, constituting the biomass of prey. And as you again get sort of into the South Lagoon, that has changed. And just from some recent investigations that Qifeng's team and my team have done under the TLM, we now have seen that the composition in the northern South Lagoon looks a lot more like what you've got sort of in the North Lagoon previously. So we've really seen now a transition into the South Lagoon that was enabled by the lowering of the salinities. So when you look at a trophic component, it's not only the overall biomass, but maybe when you are out in the shops and you have to make a choice between products you're getting, you pay attention to the kilojoules per gram that you see on the back of the packages in the fine print. And so in some ways, any of the higher trophic levels have to do, if they can, similar thing, and they can choose sort of between prey items that bring them the greatest kilojoule per gram of prey that they can forage if they can access it to the border level, and the depths of their occurrence matters. So we've haven't had any values pretty much for most of Australia at all for kilojoule values of macroinvertebrates. So we've really done some novel things here by determining how much energy content there is for all of these prey items. And we've done that with a machine called a calorimeter. And so that then generated the data that you see on the plot here. So all of this is per 1 gram dry mass, and it shows you how many kilojoule are in there for detritus, plankton, the rupia, and the algae, and then you go into a variety of different macroinvertebrates, and then a variety of different fish. And so, and as we know already from what Qifeng said, also a lot of the fish are eating on the macroinvertebrates, but yeah, when you've got macroinvertebrates and fish, they really have got much more kilojoule per gram dry weight. And it's not only that, so when you've got to make a choice between eating a muffin or a quiche out there, so you might also consider the different ingredients in it and how much protein or lipid they might gain you. And so we've also done some analyses where we've had enough material, so we were a bit constrained with how much dry mass material we had there. And so we then got analyses done for lipid and for proteins that you see plotted here. And again, it shows you that then the macroinvertebrates and also the fish are having much higher protein and lipid values, so they are providing really valuable food in there. And so we do need to have a system where salinity is low enough for having all of these components in there. So that was really quite groundbreaking that we finally got these values for the bioenergetic data. And then bringing the kilojoule together with the biomass data that we had allowed us to also plot graphs on the energy density. So that was, again, values that were of relevance for the model later on. And so on these plots here, you've got the estuary on the left side, and then the north and the south lagoon. And so that shows you per unit area, per 100 square meter, the energy density in kilojoule that you've got there derived from either the macroinvertebrates or the fish. And so really documents that the lower salinity conditions in the estuary and the north lagoon are much more favorable for having and supporting the food web than the South Lagoon did. And it also shows that in the South Lagoon it really is only then the hardy heads that are constituting the energy density, whereas in the other sections you've got a much more varied component there of different groups, and so much more choice also in supporting different ways of feeding for these higher trophic levels of the birds and the fish. So that information was then all further funneled into the other working group by Simon Goldsworthy and Ryan Baring, who did the modelling. So previously, as Qifeng said, we had a conceptual understanding that she had shown before, but we never had a quantitative model, nor any model that could be used for further simulations to be done by the managers. So having all of the quantitative data on the abundance, the biomass, and also the additional information on who feeds on what in there, the modelling team could then really sophisticate that, so you can't even read anything of that, so they've identified a lot more different trophic groups in there, but it pretty much goes from the plant and algal matter to the macroinvertebrates, and then higher up to the different levels of birds and fish in there. And so we've now got some quantitative understanding on what some of these linkages are. This plot in there is for the North Lagoon, I should say. And they could also do some initial simulations and understand how a lot of that composition of the food web is subject to some of the main drivers. And again, they identified water level and salinity as being most crucial for it. And that's not much of a surprise. But yeah, so we've now really improved our understanding a lot, and it's going to be really exciting to see further how then the resilience of the food web is now really shaping up after the big flood event that we've just had. Yeah, so that's pretty much taking us to the end. It's been a huge team effort. You see a lot of names listed there from SARDI and Flinders Uni, and also thanks to Adelaide Uni for supporting a lot of the work. So we've been out in the field a lot, and we've done a lot of lab work, so it's been a massive exercise. And of course, thank you also to all of the funding bodies that enabled this.